Okay, thank you so much to John and the committee for inviting me. It's been a great pleasure and an honor to be here. So I also was encouraged to speak about something that everyone can be interested, isn't it? And so I decided to speak about energy and metabolism. You know, energy is one of the most important things in the world. I mean, besides biology, even the car industry, you know, and everything, isn't it, is interested in energy. Since energy is in very little abundance and in the world, so it's sort of very important, isn't it, for you know, for making cars, hybrid cars that can be highly efficient, isn't it, and also use very little energy. I can tell you that happened to the brain. So this is the history of the brain, isn't it? That the millions of years ago, isn't it? We have to go from this brain to this brain, and certainly the whole trajectory to this was highly depending on energy in terms of, isn't it, making a brain extremely efficient in terms of energy and also, I mean, as I said, low energy use. And for this, there was extensive gene reprogramming throughout this time, isn't it? So basically, the main idea, a lot of the genes that we have today, the genes are highly dependent on energy. So that's why, isn't it, when we are talking about Alzheimer's disease, for example, isn't it, they said the diabetes and a lot of these issues, isn't it, is something extremely important, isn't it, for the biology, because the genes are highly dependent on energy, and any problem actually strikes on this. And you know already that the human brain use a huge percent of the total energy of the whole body, isn't it, in spite of the size. So so basically, I am going to be talking about this, and I am going to be also discussing in terms of the effect of fructose. You know, fructose is a sugar that is highly consumed in the Western society. And we have been using fructose because of what it does actually promote metabolic disruption to the brain. I am going to be talking about this. I am going to be talking also a lot of these effects when we are talking about metabolism. There is a very important involvement of the brain-gut axis. And I am going to be saying a few words also about this new science of nutrigenomics, isn't it, that it's a big data science, and how this can derive a lot of information, isn't it, for the future. Basically, this is the diagram that I am going to be going around, isn't it, but this is the main idea. So when I am talking about energy, here we have the mitochondria almost at the center. So the mitochondria supposedly is working normally most of the time, isn't it? However, can be highly stimulated by diet, isn't it, like fructose or a high-caloric diet, particularly when we are talking about free radical formations. Actually, we are talking about the mitochondria, isn't it, when the energy production is not working well, and this can turn into lipid peroxidation. And these also are territories of the foods, because a lot of the foods, when we are talking about omega-3 fatty acid, for example, or other fatty acids that we were discussing yesterday, we are talking about breaks in the plasma membrane, isn't it? Because actually these are components of the plasma membrane, and actually they provide fluidity to the membrane. If for any reason these are not working well, there are many receptors which are embedded in the membrane, and when the membrane is not working well, the signaling is going to be affected. And when the signaling is affected, I mean, practically everything can go wrong, isn't it? We can be talking about the brain, we can be talking about the retina, whatever. there will be problems in the electrophysiological activity that is being conducted, isn't it, across axons, for example. And it's going to be a vicious circle that is certainly problematic for brain function. So in terms of energy, you may know about this already. There have been many studies, and it's very well known now that the bioenergetic status is a major predictor, isn't it, of health. And there are many studies in many different species, isn't it, going from yeast to humans. For example, the effect of the thyroid restriction, isn't it, that reducing the overproduction of energy seems to increase, isn't it, lifespan. Also, we know that the beneficial effect of controlling this energy in terms of reducing the number of calories, isn't it, and it has many effects, isn't it, even in humans. And when that doesn't happen, a lot of disease happens, isn't it? So like we have been talking about Alzheimer's disease, diabetes, cancer, no question, isn't it, is very highly dependent on when this goes wrong. So basically, I think it's very important, isn't it, to think about this, isn't it, when we talk about Alzheimer's disease or we talk about the eye. When we say, for example, the eye can provide some clues to understand Alzheimer's disease. I mean, these are the common rules in the body, isn't it? Everything is connected, and there are certain conveyors of all of these, what happen in the brain. So I am going to be saying a few words about fructose. As I said, the fructose has become extremely relevant in the last 20 or 30 years because it's present in many of the beverage, desserts, and everything that we consume. One of the problems of fructose the people didn't predict when we started using fructose. The people started replacing glucose for fructose because it's sweeter, isn't it, cheaper and everything. But it's known today that the metabolism of fructose actually is different to glucose. And one of the problems of fructose that the metabolism is different, isn't it? And for example, in the liver, the fructose goes quickly to production of fatty acid, triglyceride, that you may know, isn't it, that these are very problematic, isn't it, in terms of promoting inflammation and other things. So that's one of the evils of fructose. And I'd like to tell you a few things that we have done in order to understand better the effect of fructose, isn't it? One of the things was to report this metabolic syndrome that we normally think is just a property of the body, also occurs in the brain. And for this, we did some studies years ago that in terms of the connection between diet and metabolic syndrome, basically what we did was to have animals that they were put into diets. They have some basal diet in high omega-3 fatty acid or deficient in omega-3 fatty acid. And on top of that, we were feeding the animal fructose, 15% fructose in this case. So the only thing what I want to show you here is that when we look at the levels of insulin resistance, for example, in the body, the worst condition was when we were combining this deficiency in omega-3 fatty acid and fructose. And this, the high omega-3 fatty acid was sort of protecting against the effect of fructose, which actually is a quite interesting situation, isn't it, in terms of the combination. But perhaps the most interesting for us those days was to find actually that the animal that developed this high level of insulin resistance, they had more difficulties to learn attacks in the Barnes maze. So then we continue with these studies in order to characterize, isn't it, what could be the effect of the fructose. And we publish many things, isn't it? These are just examples. For example, you may know that the long-term potentiation is one of the electrophysiological bases of learning and memory. And we look at, for example, TP in the animals, and we found that the animal being fed fructose had a reduction on LTP and also have an increase in long-term depression, which is the opposite to the learning. There were problems in it with the synaptic cleft. And also, I want to emphasize here, isn't it, that a lot of these effects, metabolic effects of food, omega-3 fatty acid and everything, are perceived at the level of the synapse, because you may know, isn't it, that the omega-3 fatty acid are part of the membrane, as I said, and everything which is embedded in the membrane is going to go wrong if this doesn't happen. So anyway, this is a very common pathway for the effect of fructose, and we saw many other effects, isn't it? This is more, a little bit more details in terms of the effect on the plasma membrane. Here we measure some of the markers, which is for HNE, which is a marker for the lipid peroxidation. Actually, is a main effector of the negative effect of lipid peroxidation on the membrane. Basically, what we found, what I told you again, isn't it, that the animal that they developed this problem with the lipid peroxidation of the membrane, when we fed the animal fructose under low levels of omega-3 fatty acid. So anyway, as I said, the plasma membrane is extremely important, isn't it, for all of these functionality of the brain, the retina, and anything that you name. We did a lot of work on the mechanism of this. I'd like to show you a few things in order to, so we try to test all of this diet, isn't it, in a model of traumatic brain injury. And for this, we have been working with a model of concussive brain injury called fluid percussion injury, which is a model of boxing or football, isn't it? Now it's more relevant these days. But basically, there is not a direct damage to the brain. It's mostly external damage. So as I said, the main denominator of all of this, I think, is metabolism. And you may know about this also in many diseases, like Alzheimer's disease. It's known that there is a mitochondrial function and everything. So basically, when we combine a system that it has a dysfunction in metabolism, like in this case, for example, traumatic brain injury, the brain, isn't it, after receiving a traumatic brain injury, goes into a period of metabolic dysfunction. So when you combine, for example, like an agent in the diet that reduce that increase the dysfunction of the mitochondria, the situation can be very problematic. And we published several studies showing this, that fructose aggravate most of the effects of traumatic brain injury on the brain. So I am not going to go over those studies. I'd like to show you some new stuff that we have, which I think is quite interesting in order to understand, isn't it, these effects of the food on the brain or retina. So this was quite interesting because actually what we did was to do a traumatic brain injury to the brain, and we look at the liver. I told you already that the fructose is known to increased triglyceride formation in the liver, which is a problem. Triglyceride can go to the brain, can be related to many disorders like Alzheimer's disease and many other. So, and that's what we found basically, isn't it, that when we look at the liver of animals being fed fructose, there was an increase of lipid droplets in the liver. But the most interesting of this is when we combine this traumatic brain injury with fructose, there was a huge increase of these lipid droplets in the liver. So it's a quite interesting story, isn't it, how the brain, something very important to think about this, that the brain is connected to the periphery, many organs, through the hypothalamic pituitary axis, also autonomic nervous system. And basically, according to this model that we were combining TBI and fructose, we have these lipid droplets formation, isn't it? And we measure several other things, but basically the main idea that a lot of these effects that come from the liver actually went to the brain, and that already is also a lot of these molecules that we are talking about, the brain in Alzheimer's disease, like amyloid, and also they are made in the liver. So anyway, I like to switch gears a little bit. And I would like to go to the future right now. Isn't it one of the problems when you study food is we have so many signals, isn't it? And and particularly in this case, for example, if we are consuming a nutrient, like a broccoli, for example, and we decompose this, you can see vitamin C. It's like a sort of pharmaceutical compound. Spinach, for example, this is the composition of spinach. So you can imagine if you want to really understand the mechanism of this, it's so difficult because we have so many components, and that also has been a really bad word for NIH study sections, because when the NIH review grants, they want to know a specific mechanism of related to specific things. So anyway, I'd like to call your attention this about data. Isn't it? We had this article written in The Economist, and last year, isn't it? And actually, it's so interesting, isn't it? Because the world's most valuable resource is no longer oil, but data. So basically, everything these days is around data. The idea is I need to understand things, and we have all of these companies making connections around the world and everything. and the same thing with biology. If for any reason we want to understand, for example, the effect of diet on cell metabolism, this is just a small summary of the reaction that happen in the cell, which actually is completely crazy, isn't it? Try to understand this. We may know partially one of them, isn't it? But very difficult to have a whole idea of this. So anyway, we start working on doing genomic analysis, isn't it? Trying to analyze thousands of genes being affected by food. And I'd like to show you just a summary of some of these. For example, looking at the high fructose effects, isn't it? Looking at different regions in the brain, hypothalamus, hippocampus. So basically, as I said, if you just use fructose, it's incredible the amount of genes which are regulated or downregulated by the effect of fructose. You can actually make sense of this, isn't it? You can identify these genes and trace pathway. And this is one example, isn't it? The epigenetics, in epigenetics, isn't it? Which is actually one way, isn't it, to save information in genes. DNA methylation is one of the most stable forms of epigenetic variability. So anyway, fructose is amazing, isn't it? It can change hundreds or thousands of genes. in terms of hypermethylation or hypomethylation. And actually, in each of these cases, basically, what methylation is doing is to regulate gene transcription of genes, isn't it, in terms of making proteins. So anyway, it's a lot to learn about this. And in order to make sense of this, so you can use the same type of analysis. For example, in this case, isn't it, we publish like the effect of fructose DHA, and we have published data, isn't it, like, for example, DHA can counteract some of the effect of fructose in terms of triglyceride formation, insulin behavior. You can use the same system biology, isn't it, to construct pathways like this, and you can identify master regulator genes which can regulate many genes related to this. For example, we found this gene FMOD, isn't it, that it happened that respond to the effect of the fructose, also respond to the effect of DHA, and also some blockers. So basically, you can use the system, isn't it, in order to design interventions. And also, you can connect this to genomic-wide association studies, isn't it? Connect these studies that you do in rat models of diseases. You can connect it to human libraries, isn't it, in terms of Alzheimer's disease libraries. And so you can know, for example, the type of risk factors that you are describing in your animal model can be related to all of these disorders, isn't it? And basically, you can connect your results in animal models to humans. And this is the last slide to show. And this is some new study that we are doing, which actually is extremely exciting. One of the most exciting questions, I think, these days, isn't it, is the individual variability. As you know, isn't it, for example, different people have different susceptibilities and need to get Alzheimer's disease, and some people can have some people, isn't it, can eat a lot of fructose or calories and get thinner. And some people, it's just the opposite, isn't it? You just eat a little ice cream, isn't it, and you gain a few pounds, isn't it? And so it's very frustrating. So the same thing happens, isn't it, in terms of treatment design, in terms of diagnosis and everything. And so we start working in a model with there is a professor, Jake Lucis at UCLA, who developed something called the diversity panel, which actually is a model in mice that he characterized 100 mice genetically. And basically, so you can use these mice as a model to understand people. And that's what we are doing right now. So for example, here, these are 3 strains of mice, B6, DBA, and BB, that were exposed to the same treatment, which was fructose. And it's amazing, isn't it? Because the type of genes that are upregulated in each one of them is completely different. So anyway, it's quite interesting, this, isn't it? How different can be the body, the brain, isn't it, in terms of the reception to the same stimulus. Anyway, we need to understand more that, and I'd like to complete my talk, isn't it? I think I already explained the effect of diet and plasticity and function, the bioenergetic importance in all of these, how big data can be important to understand this, and precision medicine implication. As you know, around the world, there is a big push to have precision medicine being developed properly, isn't it, to treat patients in terms of the individual basis. And I think I explained this already, and these are the people who helped me with these studies, and a lot of these studies have been funded by the NIH. So anyway, that's all what I have to say. Thank you.
This session's open for questions. Sure. I can get us going. I had a question for you. Could you speak to the direct mechanisms by which fructose may then affect brain and cognition? I mean, one thought would be neurogenesis and maybe some modulators there. I don't know if you had any comments on that.
As I said, one of the main mechanisms that fructose can affect is through bioenergetics, isn't it? That's what we have found, isn't it? One of the first findings is like affect mitochondrial function, and we have measured many markers for mitochondrial function, and all of these markers also happen to be related to other processes, isn't it, in terms of regulation of learning and memory and everything. So the most striking, as I said, isn't it, fructose, as you said, isn't it, can affect neurogenesis, can affect so many plastic events in the brain and the body, but as I think the main mechanism again is cell metabolism or bioenergetics.
Thank you. Any other questions?
Thanks. It was a great talk. I was just very curious to know on the last slide on the Venn diagram, what was the single gene that was a triple overlap?
The single gene?
Yeah, there was one gene that was listed as overlapping all three strains.
Yeah.
Which is sort of tantalizing.
Yeah. So we are not listing genes here. These are pathways right now. But we are identifying these genes actually was the F21, FGF21, that we found that it was a very, that's the what you're asking, yeah, FGF21. Yeah.
Okay.